Before we begin, I would like to respectfully acknowledge that we're located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples. Catch my breath here. Including the Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, and Anishinaabe, Inuit, and many others, whose histories, languages, and cultures continue to enrich our vibrant community. I am delighted. This is one of my favorite rounds of the year. I know I say that about many, but it really is a lot of work moving through promotions and that whole period of time where we gather things. And so we want to highlight our professors, and this is one of those rounds. So Dr. Paul Forsythe obtained his PhD in immunopharmacology at Queen's University, Belfast, UK. Not the Queens that we have here. He then went on to pursue postdoctoral studies at the University of Alberta and McMaster University before becoming a faculty member in the Department of Medicine, McMaster University, and principal investigator at the McMaster Brain Body Institute. He joined the University of Alberta as the AstraZeneca Canada Chair in Asthma and Obstructive Lung Disease in 2021 and co-director of the Alberta Respiratory Centre. He's also the specialty chief editor for Frontiers in Neuroscience, Gut-Brain Axis. His research interests are in the area of psychoneuroimmunology, utilizing interdisciplinary approaches to investigate crosstalk between components of the nervous, immune, and endocrine system. His work has provided key insights into how nervous system and immune system act together to provide microbiota-induced changes in the brain. In his current research, Dr. Forsythe is investigating microbial and other environmental influences on neuroimmunoendocrine interactions in the airways, the contribution of such interactions to lung disease, and how this knowledge can be exploited to develop novel therapeutic strategies. So his presentation today is on psychoneuroimmunology of the Holobiont, integrated science for integrated medicine. So over to you. And again, I apologize.
Thank you for the breathless introduction. And I'm very happy to be able to present Grand Rounds for the first time. Just to start, so first of all, unfortunately, I have no conflicts. So psychoneuroimmunology of the Holobiont. As you'll see in a few moments, that title really gives me broad scope to talk about a lot of things. But what I do want to do today is, first of all, introduce some concepts related to that title, and then use sort of three different stories from research I've been involved in to introduce the idea of first of all, microbiota gut-brain communication, which is been a focus of my research for about 15 years now. And then discuss some more recent research, really looking at the lung as a sensory organ. And then finally discuss some things about lung-brain communication. So psychoneuroimmunology. So this is the area that most of my research falls under. It's a branch of research that really studies how the brain, nervous, and immune systems interact with each other. And traditionally, it's been involved in addressing questions such as how does stress or psychological stress induce illness? And then on the other hand, how does peripheral inflammation impact things like mood disorders? For a long time, it was a very niche area of research and sometimes viewed a bit skeptically, but that's definitely changing more recently. So a sort of central concept of psychoneuroimmunology is the fact that in the body, the optimal fitness and defense against external threats is maintained by the coordinated action of the nervous, immune, and endocrine system. And while traditionally these systems have been studied in isolation, sort of in different scientific disciplines of immunology, neuroscience, and endocrinology, in reality, they're in constant bidirectional communications and they act together. To such an extent, this really has to be considered as a neuroimmunoendocrine supersystem. And what's emerged in maybe the past 20 years is that our commensal microbes are fully integrated into this neuroimmunoendocrine supersystem. They influence the function and the development of the individual components of this system, and they can also influence the communication between the components of the system. And the sort of level of integration is such that really we have to consider ourselves as a multi-species organism or a holobiont. So a holobiont is a term used to describe a co-evolved unit of the host and all its associated microorganisms. The holobiome is a term used to describe the union of all the genes that are contained within that holobiont. And really what it does is describes an interdependent symbiotic relationship between the host and the microbiota. And we know that our commensal microbes provide the host with protection against pathogens, although they also provide nutrients. But importantly, they're involved in the normal development of the immune system and normal development of the nervous system. And there's also now very clear evidence that they can modulate brain function. And that's what I want to talk about a bit more today. But overall, it's a combination of sort of host, microbial, and viral components of the holobiont that determine who we are from a physiological point of view, and as a consequence, who we are from a psychological point of view. So we are holobionts. And when it comes to understanding mammalian holobionts, not unsurprisingly, a lot of the focus of the research has been on the gut. So this is where we have the largest number of commensal microbes and where they interact with actually the highest density of immune cells in the body. And there's also a vast network of sensory nerves in the gut. So these microbes interact with the nervous system and the immune system in the gut, and this has impacts throughout the body, including influencing brain function and behavior. And I became sort of involved in this work through actually what started out as a very simple experiment. So we fed a particular commensal organism, Lactobacillus rhamnosus, to mice. And we did this for 4 weeks. These are biopsy mice, so they have sort of a baseline high level of anxiety-like behavior in these mice. We fed the mice with the bacteria, then we tested their behavior and collected brain tissue. And what we found is that feeding this bacteria changed the behavior of the mice. And first of all, it seemed to reduce their anxiety-like behavior. So here you see the open field tests. So mice, these biopsy mice in particular, normally spend a lot of time in the periphery. When they become less anxious, they will explore this open field and spend more time in the center. And you can see that the animals that were fed the bacteria spent more time in the center of this open field. And then this is the elevated plus maze. So it has an open arm, a closed arm. Less anxious mice spend more time exploring the open arm of this elevated plus maze. And you see the mice that were fed the bacteria spent more time. So this is evidence that there was sort of an anxiolytic-like effect of feeding this bacteria. It also seemed to have antidepressant-like activity. So here's the tail suspension test. In this test, you hang a mouse by its tail. It struggles to get free and eventually gives up. And this is called behavioral despair. Any known pharmacological antidepressant will increase the time that that mouse struggles and decrease the immobility time. So you see here, fluoxetine decreases the time that the mouse spends immobile. And our bacteria actually reduce this time to the same degree as this SSRI antidepressant. So this is a bacteria, when you fed it, had anxiolytic and antidepressant-like activity. So these were interesting observations. What we really wanted to know was how this was happening, sort of step by step. How could a bacteria in the gut actually end up influencing brain function and behavior? And so we took our bacteria and really used this as a model organism to explore the pathways of microbe gut-brain signaling that could modulate mood and behavior. Now, when we start thinking about potential mechanisms, we first focused on the vagus nerve, because really this is a hardwired connection between the gut and the brain. So it's the 10th cranial nerve. It runs from the medulla oblongata right down to the colon. innervating all the visceral organs on the way. Sort of 80% of vagal nerves are afferent, so that means they're sending signals from the body to the brain, and they're critical for interoception. So this is how the brain senses the physiological state of the body and allows you to answer the question, how do I feel? And interoception is also involved in mood regulation. So there was a lot of evidence. Also, vagal nerve stimulation is FDA approved for sort of treatment-resistant depression. So there were a lot of information suggesting that vagal activity could modulate mood and behavior. So to address this, again, fairly simple experiment, we just cut the vagus nerve subdiaphragmatically. So now there's no, in mice, so there's no vagal signaling between the gut and the brain. We fed the bacteria, we repeated our behavioral tests. So in the sham-operated animals, we got this anxiolytic-like effect of the bacteria, but in those that had undergone vagotomy, this anxiolytic effect was lost. So this is just another test of anxiety over here. This is the light-dark test. Again, less anxious mice spend more time in the light. You see more time in the light with the bacteria and the sham-operated, and then this is lost with vagotomy. So this was strong evidence that you need the intact vagus nerve for the anxiolytic and the antidepressant effects of this bacteria. We also had evidence that the brain becomes aware of this bacteria in the gut very quickly. So we looked at neural activation in specific brain regions. I'm just showing the amygdala here. 2 hours after feeding with the bacteria. So we looked at CFOS expression. So this is activation of neurons within the brain. It increases 2 hours after feeding the bacteria. The brain's aware of the bacteria in the gut. But again, in the vagotomized animal, you don't get this neural activation. So again, vagus nerve, critical for brain sensing the presence of the bacteria and then inducing the antidepressant and anxiolytic-like effects. So this is work that was in collaboration with a colleague at McMaster University. He's an electrophysiologist. He has a setup where he can isolate a piece of the gut of the mouse. And from here, they have the mesenteric nerve bundle. And he can then patch clamp onto this mesenteric nerve bundle, measure the electrical activity of the vagus nerve, and then introduce components into the gut. And when we introduced the bacteria into the gut here, we saw changes in the firing frequency of the vagus nerve so that it was immediately almost detecting the presence of this bacteria in the gut. And then I'm not going to go into this in detail through a series of experiments using this kind of model. It was determined that actually the signal going to the vagus nerve has to first go through the enteric nervous system. So the intrinsic primary afferent neurons are the first neurons that detect the bacteria, and then they signal through a synapse to the vagus nerve, signals to the brain. And we also identified that you need an intact intestinal epithelium to get this response. So it looks like the bacteria is signaling through the epithelium to the enteric nervous system, then to the vagus, then to the brain. So this looks like a fairly straightforward story. Gut microbes activating the vagus nerve, signals to the brain, modulating behavior. But as I said at the start, nervous system, immune system, endocrine system, they don't operate in isolation. One of the sort of first observations we ever made with this bacteria was that it's anti-inflammatory. So we first tested this bacteria in a mouse model of asthma, and we demonstrated that feeding that bacteria actually attenuates allergic airway inflammation in the mouse. And this attenuation of airway inflammation was associated with an increase in anti-inflammatory or immunoregulatory T regulatory cells. And we went on to demonstrate that actually this induction of T regulatory cells was essential for the anti-inflammatory effect of the bacteria. Why this was interesting is there's a lot of evidence out there that T regulatory cells in the periphery are involved in regulating mood and behavior. So there's a lot of evidence that peripheral inflammation is associated with mood disorders, and that in patients with major depressive disorders and PTSD, there's actually a decrease in this regulatory T cell population. And if you successfully treat people with depression, the T regulatory cell population increases. And there's also been a few reports from animal models that suggest T regulatory cells may themselves have anxiolytic effects. So we were interested to see whether this anti-inflammatory response to the bacteria could also be influencing the behavior we were seeing. So here we took a very similar approach to what we did with the vagus nerve. We sort of removed the T regulatory cells from the situation, this time using anti-CD25 antibodies. So this depletes the T regulatory cell population. And then we fed with the bacteria. And again, we saw sort of antidepressant effects in the normal control animals. And these antidepressant effects were lost when you removed the T regulatory cells. And same with the anxiolytic effects. Fine in the isotype control treated animals. If you remove this T regulatory cell population, you also remove the anxiolytic effects. So this showed that T regulatory cells seem to be necessary for the anxiolytic effect of the bacteria. We next wanted to show were they sufficient. So in this set of experiments, we fed mice with the bacteria. We then isolated their CD4, CD25 cell population. So this contains the T regulatory cells, and we adoptively transferred them into naive mice that had never been fed the bacteria. And what we found is that the behavioral effects of the bacteria were actually transferred with these regulatory T cells. So again, here you see an anxiolytic effect of T regulatory cells from mice that have been fed the bacteria, not from those that received T regulatory cells from a mouse that just received water, a control mouse. And again, this reduction in behavioral despair activity was transferred with the T regulatory cells. So now we had a situation where we demonstrated that the vagus nerve was essential for these behavioral effects. Also, these T regulatory cells and the immune system was involved. So how was this interacting? Was this parallel? Was it in series? What was going on here? But what we do know, and there's a lot of literature now, is that although 80% of vagal signaling is afferent, there's a very important efferent function. And one of those functions is actually regulation of the immune system. And there's a vagal anti-inflammatory reflex. So you have an inflammatory signal, it signals the vagus nerve, goes to the brain. There's an efferent response that is anti-inflammatory. And part of that anti-inflammatory effect is actually the induction of T regulatory cells. So we wanted to see whether this could be going on in our system. Again, we went back to our vagotomized mice and showed that if you conducted vagotomy, fed the bacteria, you lost the ability to produce the T regulatory cells. And of course, as we demonstrated before, you lost the anxiolytic-like effect. So this looks like essentially the bacteria, it's not a straight up and down response between the gut and the brain. What's happening is the bacteria are activating a vagal anti-inflammatory reflex that's inducing T regulatory cells. These cells are then available to suppress peripheral inflammation, but also seem to have an influence on brain function and behavior. So where are we going with this? We're not really interested, or never were really interested in developing probiotic treatments, or so-called psychobiotics, but taking the sort of new biology that we learned about in understanding this gut-brain communication and seeing if we could use some of that to develop potential therapeutic approaches. And so I just want to mention electroceuticals for a moment. So these are devices that can induce action potential in neurons, modulating neural activity for therapeutic benefit. And if we go back to these experiments, we were able to see changes in the firing frequency of the vagus nerve in response to the bacteria. We can actually look at this in more detail. So this isn't coming out very well, but you can sort of break down the firing pattern of the vagus nerve and assess various parameters like, so the mean interval between bursts of firing of the nerve, how big those bursts are, what's the space between those bursts. And when we broke this down, we found there was a consistent pattern in the changes of firing of the vagus nerve. So for instance, there was a decrease in the mean interval, and then there was an increase in the duration of the bursts, for example. And this was consistent. But what was also really interesting is when we exposed the gut to SSRIs, so fluoxetine and sertraline, both of these antidepressants also seem to have the same effect on the vagus nerve, altering the firing pattern in the same way. Whereas we used a different class of antidepressants, NDRI, so noradrenaline dopamine reuptake inhibitor, it had a completely different pattern. And we wondered, whether there might be some relationship between this firing pattern and vagal dependence on the anti-inflammatory or antidepressant-like effects. And so we tested this, and actually we found that oral antidepressants in the mouse, the antidepressant-like effect in the tail suspension test was dependent on an intact vagus nerve. But when we looked at the NDRI, that actually having a vagotomized animal didn't influence the ability of this to reduce behavioral despair. So it looks like oral SSRIs, at least in this mouse model, at least some of their antidepressant-like effect is dependent on intact vagal signaling between the gut and the brain. And we were wondering, do they activate the same sort of antidepressant pathway? What's interesting is that around the same time as we published this, another group were looking at actually the anti-inflammatory effect of SSRIs. So this is fluoxetine being able to suppress the endotoxin response, inflammatory response in a rat model here. And actually they lost this anti-inflammatory effect of fluoxetine to endotoxin in vagotomized animals. So this suggested that SSRIs were also activating this vagal anti-inflammatory reflex, really showing strong parallels between what we were seeing with oral SSRIs in the mouse model and what we were seeing in feeding the bacteria. Could we mimic the effect of oral SSRIs and the bacteria just by electrically stimulating the vagus nerve? And this is sort of where we are with this project at the moment. This is still ongoing. We're in early stages. So we're doing transcutaneous stimulation of the neck in the mouse. And here we sort of have no stimulation. We do random stimulation. Then we stimulate with the code that we see when we feed the bacteria or give SSRIs. And we find that this code actually has antidepressant-like effects, similar to actually giving sertraline directly. And then when we look at the immunomodulator activity and the induction of T regulatory cells, again, random stimulation doesn't increase T regulatory cells, but code stimulation does. So this looks like we can, at least in this preliminary data, sort of mimic the effect of feeding the bacteria or giving an oral SSRI by direct vagal stimulation of the vagus. So electrical stimulation mimics the effect of the L-rhamnosus and the SSRIs. So just to summarize this part of the presentation, sort of neural and immune pathways combine to mediate microbe effects on behavior. The vagal anti-inflammatory reflex is key to this response. And we can also replicate the immune and behavioral effects of both the bacteria and SSRIs through vagal stimulation. And we think that we have a better understanding of these changes in vagal firing patterns may lead to more precise electroceuticals. So as I said, vagal stimulation is used for treatment of depression. Maybe if we can tweak the firing pattern, we can make that more effective. And we can also then use this for anti-inflammatory treatments. So I'll move on to the next part of the presentation, some more recent research. So this probably wasn't the first time you've heard about the gut-brain axis. It's all over the place. It's in popular sort of science press. It's seen as one of the really important new discoveries in medicine, etc. But actually, I don't think the gut-brain axis is that special. The brain responds to all or communicates with all organs in the body, and a lot of those organs have a microbiota. So you have then a lung-brain axis, you have skin-brain axis, urinary tract brain axis, and all of these organs also communicate with each other. So now you end up with an axis of axes, and But what it really is the holobiont regulating itself. And, but we've become interested in sort of how the lung and the brain communicate, or the airways in general and the brain. So I want to talk a little bit about that. So we have to acknowledge that the lung is actually a sensory organ. It's innervated. It receives input from the inhaled environment. So you have pollutants, you have pathogens, you've got change in moisture levels, pressure, oxygen levels, CO2. They're all picked up by sensory neurons, signal to the brain. And then different stimuli induce distinct responses when it gets to the brain. So you can have changes in breathing patterns. You can have things like bronchospasm or cough. So it's protective reflexes. But like the gut, these sensory neurons don't generally project into the lumen of the airway. So actually the epithelium plays an important role in transducing signals from the environment to these sensory neurons. And there's one particular cell within the airway epithelium that seems to be important, or play an important role in this. And these are the pulmonary neuroendocrine cells. So they're rare, specialized epithelial cells in the conducting airway. They're neuroendocrine cells, so they contain dense core vesicles with a lot of neurotransmitters and neuropeptides in them. We know that they're evolutionarily conserved, so there are equivalent cells actually in the gills of fish. They're also the first sort of differentiated, mature epithelial cell to appear in the airway during development. So this evolutionary conservation and the early appearance suggests they have an important physiological role to play. They can be found individually throughout the airway or clustered together in what are termed neuroepithelial bodies. And these neuroepithelial bodies are found largely at the branch points of the airway. And so that is thought to put them in a good position to detect these signals from the inhaled environment. They are chemosensors, so they have receptors that can detect hypoxia, things like nicotine, mechanical stimulation. They also express olfactory receptors, and they can respond to volatile chemicals. When you activate these cells, they release their dense pore vesicles. But importantly, these are some of the only cells within the airway epithelium that are directly innervated, and they're innervated by sort of vagal and non-vagal sensory neurons, and they have a direct connection to the brain. So this is a cell in the airway lumen that's directly signaling to the brain. And also in a number of lung diseases, you have either an increased number of PNECs or you have increased secretory activity of the cell. So asthma, COPD, CF, bronchopulmonary dysplasia, and bronchiectasis, they all see increased numbers of these cells. So they seem to have a potentially important role in pathology, as well as being important in signaling to the brain. So these cells are important chemosensors involved in lung-brain communication. The problem is they're very rare. So over here you see single-cell RNA sequencing of human airway epithelial cells. The total population of cells, this is the population of pulmonary neuroendocrine cells. Okay, and there are no suitable isolation methods. So we don't know a lot about these cells, especially in the human, because we can't isolate them and study them. But we wanted to learn more about these cells. And so we decided we would come up with a model system to study them. And this is some work that's been conducted by a postdoc in my lab, Rachel Nuttall. And the first approach we took was to try and develop these pulmonary neuroendocrine cells from induced pluripotent stem cells. This is a method that had just been published a few years ago. You basically take your pluripotent stem cells and you take them through a number of stages, eventually into lung progenitors, and you then put them in an air-liquid interface, treat them in a certain way, and you start getting enrichment of pulmonary neuroendocrine cells within that cell population. That takes 6 different media, a whole bunch of growth factor cocktails, and about 90 days to get a culture that you can actually use. But there was some evidence from the literature that actually PNECs could differentiate from airway basal epithelial cells. So actually, if you expose basal airway epithelial cells to hypoxia, some of those cells start to turn into neuroendocrine cells. And it's known that actually it's notch signaling from adjacent cells that prevent PNEC development. So we thought if we took human bronchial airway epithelial cells, we inhibited notch signaling, we might get some of these pulmonary neuroendocrine cells developing. And so that's what we did. We took human bronchial epithelial cells, we inhibited notch signaling in this airway epithelium, we put them at an air-liquid interface, and we cultured the cells. Here you only require 2 culture media, but it still takes 60 days to start seeing pulmonary neuroendocrine cells appear. Then we characterized these cells looking at sort of classical neuroendocrine markers, gene expression. So we have the IPNEC culture, this is the pluripotent stem cell, the EPNEC, that's our epithelial-derived pulmonary neuroendocrine cells, and then the non-enriched normal human bronchial epithelial cells. And you start to see the emergence of all these classical neuroendocrine markers, so synaptophysin and chromogranin, which are canonical markers of neuroendocrine cells, appear. They're not at all present in the human bronchial epithelial cells. And then you also get things like the gene that encodes for substance P. So not just that we had this gene expression, but also these cultures released substance P into the media. And at the time, this was actually the first demonstration that any of these culture systems actually acted like neuroendocrine cells rather than just looking like neuroendocrine cells. And then we looked in our culture using single cell RNA sequencing. And so we had about 20% of our airway epithelium culture was now pulmonary neuroendocrine cells. We had 2 populations. So we have a population here that's really the mature, we think, pulmonary neuroendocrine cells, and these other population are on their way to becoming neuroendocrine cells. There were also 2 populations in the IPNEC culture. But when we looked at what other cells were in the culture, in the IPNEC culture, from the pluripotent stem cells, you got a whole mixture of cells like fibroblasts, smooth muscle cells, and then cells that were left over for each of those stages of development. Whereas when we looked at our epithelial-derived cells, the other cells that were in the culture were really other airway epithelial cells. So basal epithelial cells, club cells, sort of goblet cells, that kind of thing. So we think our model really better represents the airway epithelium that's enriched for pulmonary neuroendocrine cells. And so next sort of stage of this was to see how they responded to stimuli. So we first set out stimulating them with a volatile chemical, sort of endotoxin component of bacteria, and also a common aeroallergen, house dust mite, just to see how they responded. So you don't have to pay a lot of attention to this, but just to say that we got changes in gene expression that was dependent on the type of stimuli that were used. But what really caught our eye here was that this particular gene, this is CALS-B, so this is a gene that encodes for a neuropeptide, calcitonin gene-related peptide. It wasn't present at baseline in any of our cultures, and it only appeared when we exposed the cells to house dust mite, not to any of the other stimuli. When we were able to show again, it wasn't just gene expression, but that these cultures then started producing and releasing calcitonin gene-related peptide. Why this was really interesting is there were a paper in Nature in 2018 showing that pulmonary neuroendocrine cells were essential for the development of allergic airway inflammation in a mouse model. So you're deficient in PNEX, you no longer get an allergic response to an antigen challenge, including house dust mite. And they went on to demonstrate that the actually pro-inflammatory effect of pulmonary neuroendocrine cells was mediated through the release of calcitonin gene-related peptide. And and here we were seeing CGRP being produced by our cells in response to house dust mite. How was this happening? How was house dust mite inducing CGRP from the cell? So we know that for proteases play an important role in a number of allergens. Proteases themselves can act on cells to induce a T2 immune response, similar to an allergic response. And some antigens that are not by themselves allergenic become allergenic when they're in the presence of a protease. So it's important for activity. And so we wanted to see whether protease activity had anything to do with the effect of house dust mite on our cells. And in fact, it did. So we used a broad range of protease inhibitors. If we block protease activity, we prevented the ability of house dust mite to induce this CGRP gene in our cells. So how is this happening? So there are a number of receptors that respond to proteases on cells. They're protease activated receptors, 4 different receptors. They're widely expressed in a whole range of cells. We wondered, which of these protease activated receptors could be expressed on the pulmonary neuroendocrine cells. First, we went to publicly available RNA-seq data, and this indicated that pulmonary neuroendocrine cells expressed only one of the protease activated receptors, PAR1, not the PAR2, 3, or 4. We then went to our culture, our own single cell data from the ePNIC culture, so this PNIC population, and we found the same thing. It heavily expressed PAR1, but didn't express any of the other PAR receptors. And then we carried out immunohistochemistry, and we could see PAR1 expression on the surface of our. So it was expressing this protease activated receptor. And then the next step, of course, was to use an antagonist of the protease activated receptor. And we found that this antagonist blocked the effect of house dust mite. And then if we use an agonist just at PAR1, we could replicate the effect of house dust mite. Now, this was very strong evidence that house dust mite was inducing CGRP from the pulmonary neuroendocrine cells through this PAR1 receptor. So just to summarize this component, we've developed a novel model of PNEC-enriched airway epithelium, and soon after developing this model, we were able to provide sort of novel mechanistic insights into the sensory function of the cell and the airway, and show that it did respond to proteases that might and release a neuropeptide that it was involved in regulating the immune system. And then coming right back to the idea of microbiota host communication, many bacteria also produce proteases, and it's actually key to the pathogenesis of a range of organisms, including Staph aureus. And in fact, in the skin, the itch that's associated with the Staph aureus infection is actually due to a protease in the bacteria acting on PAR1 receptor directly on sensory neurons. And we also know that Staph aureus is a major cause of respiratory infections, and it can actually induce inflammation similar to an allergic response. And so we wonder whether maybe pulmonary neuroendocrine cells might be part of an early warning system for pathogens in responding to proteases. And we have tested a protease from Staph aureus, this V8 protease, and showed that it does also induce CGRP production from the cell. So ongoing studies here. We want to develop the model a bit further. The relationship between sensory neurons and these pulmonary neuroendocrine cells is very important, and we want to build that into our model culture system so we can explore those interactions. Obviously, we're interested in more PNEC biology, how they respond to different stimuli, what profile of neuroendocrine mediators to their release in response to that, and what's the impact of these mediators on other cells in the lung, particularly immune cells. And then obviously we want to see whether pulmonary neuroendocrine cells are tractable therapeutic targets. And here we're initially focusing on developing sort of lipid nanoparticles or extracellular vesicles that might be able to deliver therapeutic targets to pulmonary neuroendocrine cells, with the idea of being potentially suitable for inhaled therapies in the future. So one final quick story. Looking more broadly at lung-brain communication. And specifically, we're interested here in the relationship between lung disease and mental health. And here, asthma and mental health. So we know that there's a higher prevalence of depression and anxiety in individuals who have asthma, and this occurs throughout their life. There are some studies that suggest there's a stronger link between depression and anxiety and asthma than with other chronic health conditions. I know that's not a consistent finding. But we do know that anxiety and depression is associated with poorer asthma outcomes, poorer control, increased hospitalizations. And what we're really interested in is evidence suggesting that this relationship goes beyond simply, well, not simply, but the psychosocial aspects of having a chronic disease, that there are real biological mechanisms driving this relationship. And so recently we are using a mouse model of asthma. This is a house dust mite, chronic house dust mite exposure. You get chronic allergic airway inflammation. And in this study, we examined male and female mice. And actually, in this experiment, we mixed the asthmatic and non-asthmatic mice in the same cage. So any of the effects you see aren't due to just what's going on in that particular cage. And we got similar levels of airway inflammation in the male and female mice, slightly more in the females. But when we looked at sort of plasma circulating cytokines, we got higher levels of circulating inflammatory cytokines in the females for almost every cytokine that we measured. This isn't new. This has been reported before, and that there's a higher sort of T2 immune response in females. And then we were looking to see what was going on in the brain, and we assumed that with the higher circulating inflammatory response in the females, that this would affect the brain, and we would get a bigger response in the female brain than the male to this airway inflammation. What we saw was completely the opposite. So this is just looking at, here I'm just showing the hypothalamus. So this is gene expression changes in the hypothalamus, comparing control mice with mice that have been exposed to this chronic house dust mite exposure. In the male mice, 555 genes were significantly altered, either up or down regulated in the hypothalamus. And in the females, 15 genes. So not big changes. When we looked at behavior, again, this is just showing the tail suspension test. Males, there was an increase in disparolate behavior in the males that had been exposed to airway inflammation. Didn't get a change over baseline in the females. Not going to go into detail, but when we started looking at the sort of resident immune cells in the brain of these mice, in the males, we got an increase in microglia activation. We also got the appearance, interestingly, of activated mast cells appearing in the brain of the male mice, close to the hypothalamus, actually. Whereas we didn't see this in the female mice. And what we saw in female mice was an activation of astrocytes. And the sort of gene expression changes in those astrocytes seems to suggest that it's inducing a sort of protect and repair program in these astrocytes. So very different sex-dependent effects on the brain to allergic airway inflammation, even though the inflammation peripherally seemed higher in the females. And then finally, coming full circle, we also looked at the gut microbiota in these mice. When we looked at the large intestine, we didn't see very many changes really in either the male or the female mice. But when we looked in the small intestine, we got really a parallel to what was going on in the brain, so that the microbiota in the male mice was changing significantly when it came to sort of diversity and richness. It actually increased, whereas the females didn't change from baseline. And we looked at this in more detail. We can't say anything about causality here, but just it is interesting that one of the significant changes in the males was loss of this particular organism, Robustia ileatus or ileus. This was lost completely in the gut of the mice that were asthmatic. What's been shown for this organism before, it's actually increased in the gut of mice that are resistant to the depressant effects of stress. So this bacteria is at least associated with antidepressant-like effects. And it's also been in a number of studies shown to be reduced in humans with depressive disorders. But of course, like I say, we can't say anything about any sort of causality here, or even what direction this relationship goes, whether it's a lung-gut-brain effect or a lung-brain-gut effect. But that's still something we're looking to explore. So just in summarize, this adds to evidence that there are biological mechanisms underlying comorbidities between asthma and depression. These are sex-dependent, so really we need a sex-informed approach to understanding and mitigating the sort of neuropsychiatric sequelae of allergic asthma and probably other lung diseases. And sort of an aside, but I think it's an important point, I think the small intestine is an underappreciated or plays an underappreciated role in the microbiota gut-brain communication. It's very rarely sampled in human studies when they're trying to look for these relationships. But actually, while it has less bacteria in the small intestine, there's a higher density of the sort of microbial sensors in the small intestine. So a higher concentration of immune cells, and it gets much higher vagal innervation than the large intestine. So I think it plays an important role here, and it's overlooked certainly in human studies. So finally, take home, the immune and nervous system combine to mediate the effects of gut microbes on brain and behavior. The vagus nerve seems to be a key facilitator of these neuroimmunoendocrine system. And we think better understanding of this could lead to better electroceuticals and potential novel therapeutic approaches. Microbiota gut-brain communication is important, but things like the lung-brain axis are underexplored. And there may be benefits to lung health and to mental health if we better understand these sort of relationships and better understand the lung as a sensory organ. And finally, just a reminder, if you're addressing any question in biology or medicine, you should think psychoneuroimmunology. And so I just like to acknowledge there's people from my lab that have been involved in this, particularly Richuman Nuttall for the PNEC experiments and Shivani Mandal, who did the asthma study at the end there, and also collaborators within the inflammation group here at the University of Guelph and McMaster University. So thank you very much.
Thank you. That was very, I guess, stimulating and eye-opening in terms of all the factors that play into it. And I'm glad I didn't interfere in your question time by being late. I'm just going to kick it off and ask you about, you talked about electroceuticals and, looking at that sort of mode in vitro in terms of therapeutics. Why have you stayed away from probiotics or looking at other? I mean, there is, as you say, a lot of work being done in this area.
Yeah.
So
we've never really been interested in probiotics, largely because there are too many variables involved. So it's a living organism. It has its own agenda, if you want to say that. And when you put it into the gut, there's going to be a lot of environmental factors influencing that. And what you put in may not stay, as it is. It's going to adapt and modify. And I think that's why, actually, when you start looking at probiotic therapies, they're not that effective. I mean, it's pretty limited, and there's probably a lot of individual variation. So it's going to work for some people and not. And that's, so I think we were always really interested in looking at these pathways and could we have a sort of less volatile, variable way of actually acting on that pathway and mimicking the effects. So we talk about a microbe-derived therapeutic approaches rather than using actual microbes themselves. So,
and I mean, it's nice that you've identified the specific microbial organism, if you will, because a lot of the studies are sort of very broad and general in terms of work that's been done. So, do you have a couple online? Do you want to bring them up? Do you want to read it for those in the room?
So in humans, electrical vagal stimulation inhibits rheumatoid arthritis is recently published in Nature. Any evidence in lung disease as well. Again, not that I'm aware of electroceuticals being used in lung disease. There's because efferent vagal activity is also involved in lung function, and so it's sort of a bit more complicated in the airways. But I'm not aware of anything. I know there's some evidence that the vagal anti-inflammatory reflex is also active in the airway, but I haven't seen anything using vagal stimulation for lung disease. I think there was another.
Yeah,
Okay.
we'll get to the
Okay, yeah.
Q&A first
Okay,
and then go back to the chat.
Did you consider synergistic effects of SSRIs and probiotics, similar to what we found as adjunctive effects of drugs in IBD and the gut microflora? Yes, we've thought about that and sort of putting SSRI and probiotics together. But actually, again, what we're more interested in is whether we can make the vagus nerve or ultravagal nerve activity so that actually that might be a good adjunct to SSRI treatment. So you're kind of making the vagus nerve more responsive to the SSRI, or so we've had some discussions about that. Again, yeah, we're staying away from probiotics, I think is the thing. And I think, the track record somewhat justifies that. And that's what I think.
Do you want to address the second
Okay.
one before the one in
Okay. Antimicrobial drugs are very commonly used blunt instruments impacting microbiomes. Any comments about the real or potential impact on either the lung or asthma and the brain and mental health? Yes. So, so I've just trashed probiotics, and this is where I think they might have some benefit. So we've done studies in the past, looking at early life antimicrobial treatment and actually using sort of clinically relevant doses in mouse models. And very definitely, if you give them sort of prenatal or early postnatal penicillin in mice, that when they become adults, their behavior is different. So we've shown that, and actually, when we fed this particular organism early in life, it prevented those behavioral changes later in life. So definitely, antibiotics used early in life have the potential to impact sort of brain development and behavior. Yeah.
Well, it's interesting because in patients that present with asthma, it's not uncommon for them to have antibiotics thrown at them, right? So you have to think about the interaction. Adrian had a question.
The carriage of rhamnosus is very low, isn't it? So two things. How is its impact on mood discovered? And secondly, you said that you can detect changes within 2 hours of induction. What's known about the bacterium epithelial interface that's causing that? You elucidated the house dust mite protease. But what happens with the rhamnosus?
So yeah. So how was the mood effects? We were interested in the potential that the gut microbiota could influence mood and behavior. That particular organism we had already shown had anti-inflammatory effects. It also reduced visceral pain responses, so that was shown before. And we thought, okay, so this regulates the immune system, it regulates the nervous system. If there's going to be an organism that might do this, it'll be this one. So we treated it, and that worked out. Yes, so the brain becomes aware of it quite quickly. We know that this signal requires the intact airway epithelium. We think it might, and there's a little bit of evidence that this might involve actually serotonin 5-HT production by the airway epithelium, and this might also explain why SSRIs have a similar kind of effect. So there's serotonin signaling from the gut epithelium to the enteric nervous system and then to the brain. Yeah.
I think we have a quick minute for the final chat one, and then we'll move on from there. I think you had.
Can you stimulate the biggest nerve in humans with yes, you can. And so this is transcutaneous vagal stimulation in humans. There's a lot of work being done now with sort of neck stimulators and that kind of thing. What I don't think they're doing is sort of tailoring the firing pattern for the effect they want to see. It's just a very general stimulator, but you can do it. And also in the ears, so there's sort of earpieces that'll stimulate the vagal nerve in the ear as well. So that's another approach that's being taken. So yes, and that's where we would like to eventually take this if it all works out.
Well, thank you. And on behalf of the department, congratulations. And thank you for a illuminating presentation and really well received.